Okay, so can we make life better on Earth by bringing life to Mars? I'd like to say that this program is dedicated to my son, Kevin Gardiner, who was born on Tuesday. You know what Tues is a German god of equivalent to Mars, right? And I always wear my Mars Society hat on Tuesday. And so he was born on September 12th, Tuesday, 1989, and he died September 30th, 2014, at the age of 25. And I'll have a little bit more to say about that later. So how can we possibly survive a 3-year round-trip mission to Mars unscathed? Now, we've identified problems in Earth orbit on the ISS, but now in 2015, the NASA Inspector General came out with this report, which said there's a lot of problems we haven't dealt with yet. 33 identified risks and 317 gaps. Leave it to NASA to come up with make a few problems into a whole bunch of problems. But we're looking at a particular level of detail, of course. So, but here's the levity of the solution. Many crew members and many solutions, and you're all crew members now, okay? So basically the approach that everyone agrees we need to do for life here on Earth and going to Mars is to reduce stressors, reduce environmental toxins that we've already been exposed to, which reduce our capacity to deal with new things and new problems, reduce radiation exposure and protect against it, watch what you eat, and exercise adequately to maintain normal respiration. Sorry, I get close to that. So here's a scorecard for the healthy solution to go to Mars and living on Earth today. Bone demineralization and calcium loss has been solved. Not without some difficulty, though, because NASA reports that the advanced resistive exercise device that the current crews use on the ISS is almost completely, if not completely, effective for reducing the bone loss. So now, going to Mars, if this is the only solution we have, you'll be exercising 2 hours a day. So maybe you ought to practice more today doing your push-ups and your pull-ups. Okay, but they've also identified a sensitivity to toxins, free radicals, we call them. I'm a chemist, by the way, and so I may throw out some terminology and raise a finger if you don't get something you want to find. I'll try to be explicit. So we have to establish a primary baseline status for studies of our homeostatic systems. This is a term doctors use to describe how we maintain good health, homeostasis, which infers, when you get out of balance, get back into balance. And in recent years, they've also identified ophthalmic changes, eye changes in astronauts. They have a loss of vision, as much as two diopters loss in visual acuity, wool spots, pressure to the back of the eye, edema in the eye, that sometimes has not been resolved after returning to Earth. The known risks of unshielded radiation in deep space, we think, are fairly well understood. And Dr. Francis Kuznada at UNLV has researched this for years, and he's done a lot of research about it. And the two main forms, as you may be aware, is galactic cosmic rays and solar particle emission events. But now we have solutions underway that may reduce radiation shielding requirements. And there's work in progress. A report we received at the National Space Society conference this year in May was given by the Sinclair Laboratory at the Paul F. Glenn Center for the Biological Study of Aging at Harvard Medical School. They've initiated a consortium for space genetics. And as you see, to protect human health, promote exploration, to sustain our planet. This only showed up last year. I've been searching for some established medical institution to put this out as something, as an approach they're taking, and finally it showed up last year. We formed our committee in the NSS in 2007. The people who are studying aging should be studying the problem of going to Mars, which Harvard is doing now. Johns Hopkins is doing it. We have a committee in National Space Society. It's got medical researchers from Columbia University, Wake Forest University, and Baylor Medical School. So Carolina Kwolek came and spoke about this. You'll see several slides here that she's she's allowed me to copy and present to you today. They've identified several components as hallmarks of aging, which they are now relating to all the problems of health and spaceflight, which includes epigenetics, cell communications, diet, nutrition, mitochondria function, cellular pathways, and epigenetic effects. She talked about all these, but I'm going to pare it down to just a few that are interesting. So and here's a bit of biochemistry. Now, everything in the body requires electron transport, which means you have to have a reservoir of electrons to make everything go. It makes enzymes work. It means protein synthesis, energy generation, all depends upon the availability of electrons, because you got kind of a downhill process, and then we've got recirculation cycles to return those electrons back to where they're useful. So, for example, nicotinamide adenine dinucleotide, the NAD plus, here is electron poor, but it is oxidized to another compound, NAM. And they have found that in breast cancer, for example, there's an enzyme that has been cut out of the genome in the cancer patient, and that's with all these little DBC1s and so forth. And as a result, this cyclical process is interrupted. And the enzyme on the left here, if it's not functional, that ends the NAD, which is the source of the electrons, is depleted, and cell death occurs. That's why you have your little friendly Grim Reaper over there. And they think that DBC1 is this enzyme that is robbing that system. The lack of that enzyme in the cancer victims is causing the depletion of NAD. So. But now we've had this new innovation that they found at Harvard, and it's been known for a few years now. So you may have heard of it, which is this nicotinamide mononuclear nucleotide, which provides a protection against the age-related DNA damage. It's an electron donor. It puts back the electrons that have been lost as a result of this cancer derangement. And so this is the effects in the cytoplasm. So the effect of that is they've actually done radiation studies at Harvard using infrared. I mean, IR is infrared, and you can see on the left that there is measures of hemoglobin on the left. It's normal with no IR. Then VEH, that's a form of the mouse they use. These are mouse studies that they did at Harvard. But I will tell you that Carolina is using NMN, and people are using this as an actual supplement because it seems to be quite a common problem. So in this chart, you can see that with the loss of the NMN, you have a loss of hemoglobin, and that's very common in a lot of disorders. Like you may get an H1B test done. That's a measure of how much of your hemoglobin has been damaged by attachment with glucose. Now you add the NMN back and maintain the radiation, and the hemoglobin levels go back up again, you see? So as I mentioned, there's a commercial application of this nicotinamide mononucleotide. I may have mispronounced it, but that's what it is. Okay? So over time, you have other complications, such as this mitochondrial genome the mitochondrial genome they see has been damaged, and so it can no longer properly generate power for the cells in our body. So we use oxygen to produce most of our energy. It's very efficient. It produces ATP, like 32, 36 ATP molecules. The other process is fermentation, which uses just glucose, and it produces far fewer ATPs, 2 or 3, and a lot longer to build up energy. So you can see that dysfunctional mitochondria, and she refers to Warburg-like metabolic reprogramming, and I'll talk about that shortly. So, in fact, Dr. Seyfried, Thomas N. Seyfried at Boston College, which I think is part of Harvard now, I'm not sure, has looked into all areas of causation, including nutrition, food, and things we swallow with our food. And he's come to the conclusion that metabolic disease as a dysfunction of energy generation is common to all cancers. And so, obviously, it's a condition that you have before you get the cancer too. It builds up over years before it finally affects enough cells. You develop a tumor. So this is something I have my wife's brother-in-law is a radiologist, and he says, "You think you don't have cancer? Let me give you an MRI, because you'll see hot spots all through most people's bodies. You'll have 40 or 50 or 60 active sites where you've already had this kind of damage." The question is whether the body is dealing with the damage, which is the issue we have with space ionizing radiation. How much damage is there going to be, and can we repair it? Well, it seems it's already being, the systems that repair it are already being damaged. So as early in the 1930s in Germany, Dr. Otto Warburg identified damage to cellular energy generation as a major factor in cancer. I know what you're going to ask me. Was he Hitler's doctor? I don't know, and I don't care. Okay? So he already figured with a lot less instrumentation back in those days, I might say, and test methods, that cancer arises from damage to cellular respiration. And the energy through fermentation gradually compensates for insufficient respiration, which is a so-called oxidative phosphorylation, oxidative generation of energy. And cancer cells continue to ferment lactate in the presence of oxygen. Now, normal mitochondria would use the oxygen, but when they're damaged like that, they only use lactate, they only use glucose, and some other forms of high glycemic carbohydrates. So this is the signature metabolic malady of all cancers, of all cancer cells. Okay, something else I inserted here, which I'd found out before, is in Cell Metabolism. You can see the reference at the bottom, an article called "Life is Short If Sweet," and in that study they showed in C. elegans, a nematode that they test on, which is very similar, has very similar oxidative phosphorylation and energy generation process. They have mitochondria, just like we do, and in the presence of circulating insulin, it blocks expression of the other enzymes involved in longevity and heat resistance. So insulin it makes for a sweet life, but it makes for a short life. And they've shown that they can extend the life of nematodes just by giving them a low glycemic diet. So so you have all these stressors on the left here. Oncogenic paradox, reactive oxygen species, age, viruses, rare mutations, information derangement, hypoxia, radiation, and carcinogenesis. All this is all in the mix. But the only thing they have in common is the Warburg effect. The cells only use mitochondrial fermentation. So this results in self-sufficiency and growth signals, insensitivity to anti-growth signals, and limitless reproductive capacity. The apoptosis, which is normal in tissues to get rid of bad dead cells, isn't working. So you got defective cells that are multiplying and replicating, and all the hallmarks of cancer are a function of this dysregulation and this misproduction of energy. However, one thing that our normal cells have that the tumor cells don't have is they can use ketones. How many people have heard of ketones or the ketogenic diet? Good. Okay, because as long as the cell has normal mitochondria, it can use the ketones, and ketones are natural in the diet from fatty acids and fats. If you do a ketogenic diet, you're going to be eating a high-fat diet, healthy fats, vegetable fats, avocados, fats of that nature, not especially animal fats. And ketogenic diets are kind of difficult to interpret when you read about them because strictly speaking, it's only about the amount of carbohydrates. But really, it's more specific than that. It's about cutting down on the high glycemic ones. Fibers in vegetables and most vegetable carbohydrates are very low glycemic. In fact, they're more like fiber. A lot of them are indigestible as such. Okay? So, Dr. Seyfried has been talking about this for a good, 8 or 9 years. You may not have heard of him because you have to be your own cancer patient who often, if you're your own cancer patient, you may have gone through other treatments. Standard treatment, according to Dr. Seyfried, and you can look it up, has not gotten any improved overall effects since we started using radiation and chemotherapy after World War II. The explanation that's sometimes given is that Madame Curie introduced radiation early in the 20th century as a treatment for cancer, but cancer wasn't considered an incurable disease at that time. There was the Beard process of using pancreatic enzymes to treat pancreatic cancer. He had a high success rate. And now we have famous people like Steve Jobs, and I'm sure you have relatives. My grandfather died from pancreatic cancer. But these treatments were working for people. But, it's kind of lost in the mists of time, except that some places like the Gerson Institute here in California does use this method, and it's better to get there first because if you've had chemo, it's not as effective. That's the Gerson Institute. I don't have a slide for that. So Dr. Kwok's group at HMS indicates, surprisingly, that DNA repairs continue normally at all ages, even under today's many and varied stressors. But there is a disruption in the expression of some genes that results in the hallmarks of aging. So here's a question. What else is being studied as a driver of both aging and a human physiological response to health barriers and stress of spaceflight? Well, here's what I found that's important in answering this question. Epigenetics. Epigenetics is, it's one of those technical terms that refers to the study of control of expression of genes results in the synthesis of all structural functional components of the body, and including enzymes that promote growth, differentiation, and maintenance. And her report shows that epigenetic processes change with time. They become kind of noisy. They cause an inappropriate gene expression that induce frailty and all the hallmarks of aging. This is kind of, this is a DVD or a CD showing your genome at 20, nice and smooth and clear. Your genome at 70, all scratched up. This is her slide too. So, causes of aging may have little to do with mutations and the things we think cause mutations, like radiation, ionizing radiation, including GCR. I'm not talking about the hard stuff that lead to this epigenetic noise in the aging process. So, a loss of epigenetic resilience over time provides a link between DNA damage, gene expression, and cellular dysfunction. These special mice may be a model replicating responses to spaceflight. These are mice that are inducible epigenetic effects, mice. Okay, so let me see. Next. So the term epigenetic and manipulation of the genes, 5 minutes, okay, often is related to the microbiome. The microbiome is all the gut bacteria that you have that reside within you. I'm sure you've all heard by now. You got 10 times many of these little critters than we have of our own cells, which is trillions. So if you could string them all up straight through space, they'd extend Alpha Centauri. So there's two kinds of epigenetics. That's the endogenous ones, because the body uses its own internal systems through the one carbon cycle that turns on and turns off gene expression. So there's internal processes that regulate gene expression that are responsible for all these important metabolic and synthetic processes, including the repair ones. But now there's also exogenous epigenetics, and that's the ones in the microbiome that consist of opportunistic microbiota that intrude our interstitial and vascular spaces, changing the expression of our genes for their own growth and reproduction. That's all they're doing. And Candida, for example, exerts all these epigenetic effects they turn off genes in our own cell tissue to do what? To farm us. They're farming us. I mean, we've become a food source for them. So they're just turning on the genes that produce these cells. Now, this is not from Professor Quallick at Harvard. This is a conclusion I've drawn from literature search that that they're replicating all these tissues and they're feeding on them. So, and there's many key triggers for yeast fungus infection, which includes antibiotics, overuse of antibiotics. If you knock out your natural flora, then Candida can flourish, because what is an antibiotic? It's a chemical substance produced by fungus to take over an ecological niche. So we're actually helping the funguses, to take over our gut in some sense. So there's a problem with this process in astronaut ophthalmic syndrome I mentioned earlier. There's a loss of production of B vitamins, and there's changes in the epigenetic expression in astronauts, so that they lose the capacity to generate nitric oxide in their epithelial tissue, or it's reduced nitric oxide production, which is responsible for vascular relaxation so you can have proper circulation. So that is reduced by some epigenetic process. So it's the underexpression of important enzymes necessary for endothelial tissue function in the blood vessel. And these are all remarkably similar to the effects of Candida overgrowth. So that's why Dr. Simoncini and some other medical people around the world are treating cancer as a fungus infection, as a primary fungus infection, with the cancer being a follow-up, a sequela for a fungus infection. Okay? And we don't have much time left, but there's other things going on here too. So you're probably wondering, well, what can I do to limit my own fungal growth? Vitamins, minerals, herbs, omega-3 fatty acids, things you're probably taking, homeopathic remedies, spices, citrus oils, essential oils. There's a lot of things that help control Candida. So the question is background ionizing radiation a phantom menace? I suggest that in large part it is, because if we have all of our systems functioning right, we have a far less likely possibility to get cancer from ionizing radiation, because normally the body would take care of itself and repair the damage that is normal from all the things we're exposed to already on Earth. See? So carbs are largely involved with this. If we cut down the carbs, that's what a ketogenic diet is. That's being treated by Dr. Seafried. They're experimenting with that. Gerson Institute uses that too. They get rid of they have a little different treatment. But that's one approach to treating cancer. But also, you can do that and advance that. You don't have to wait to get cancer to do that for yourself. 2 minutes. Okay. So a hallmark of this is changes in the pH of the body. If you're going to have, if you test your urine and your saliva in the morning, you're going to have a low pH if you're already on the acid side, because that's favorable to the Candida growth. And that can lead to anything favorable for Candida growth allows the Candida to overgrow into these long, spiky tissues that penetrate the intestinal lumen and allow food particles and undigested proteins to go into the blood circulation. And that's due to loss so that causes loss in the tight junctions of the intestines and alerts to the circulation of all these things. So I discovered in trying to help Kevin in about 2007, and I found this. And you can go and get a blood cell test for $35 in the Atlanta area. And these are my blood cells after I cleaned up myself for a while. I got rid of a lot of bad stuff that you can see in this one. Now, can you tell what this is here? That's the Hubble D field on the right. I don't have time for guessing games. We're running out of time. And that's my blood. That's my before blood sample. So you can see these are all parasites in here. This thing, if it was animated, it would be swimming. There's a lot of swimming stuff in here. The Candida, the bacteria. When I saw this, I said, "Do I need an ambulance to leave here?" He says, "Oh no, it's very normal. You may have 10 more years of life before you get cancer. Just clean yourself up." Okay, so you can get this test done, and so we need to develop biomarkers and biometrics of health. And that's what restores the electron donor capacity again. Get our pH up, because high pH, a lot of electron capacity, to a limit, only up to about 7.5, 7.8. You can get into metabolic alkalosis, which is no good. You got to stay in this little range. At 7.35, 7.45 is optimum. That's your blood chemistry, okay? So you can test this yourself. You can get one of these little bottles, ion test strips that are available at most health stores, $14 or $15. Check your pH in the morning. It's very dynamic. You'll be surprised how much it changes from day to day. Urine and saliva, fasting, first thing in the morning. So, and he also, Dr. Seaver, generated this glucose ketone index, which if you're already taking a blood sample, you can get a ketone strip too, and test for your ketones in your blood. And if you have a ratio of about 1, that's about right to not feed the cancer, and to probably prevent it, too, if you start early enough. The other thing is, when we go to Mars, we'll be able to do what? We will design our habitats from the ground up, or from the Mars surface up. We won't have to bring all the glyphosate, the pesticides, the heavy metals with us. We'll be able to put it in just the way nature intended, the way we function, and we can start from scratch. And we won't even expose ourselves to all these things that cause aging in the sense that we have here. Not to say that you're not going to die someday. This is not immortality we're talking about here. We're just, living a normal, healthy life. Okay? So, I go into a lot of detail about that. But when they first got the astronauts on the ISS, they're really happy to grow things there, vegetables and things. When they got their first lettuce production, well, it wasn't lettuce, it was wheatgrass, I guess. They said, that's one small bite for man, one giant leaf for mankind. So they realized the significance of it. So, talking about that. And Kevin in his final note says, it's a journey, not the destination. So, yeah, we can go to Mars, we can be healthy there, and we can be healthy here. Thank you.